Let me give you some examples of genetically modified organisms. First example, Roundup resistant soybean. Roundup is a herbicide made by the American company Monsanto. Roundup is actually an inhibitor of aromatic amino acid synthesis. So when it is sprayed on plants, the plants cannot synthesize these aromatic amino acids and they die. So Roundup acts as a weedicide. This soybean was made resistant to Roundup by introducing genes from bacteria. Another example is Bt cotton. Bt stands for Bacillus thuringiensis. It has a cry gene from the bacterium Bacillus thuringiensis. This cotton is resistant to insects. Cry gene produces a cry protein, which occurs as an inactive protein called the protoxin in the bacterium. Once it reaches the insect gut, it is converted into the active form, which attaches to the epithelial cells of the gut, creating holes in the gut. The insect cannot feed anymore and it dies. Bt cotton is mainly cultivated in Andhra Pradesh in India. Another example is a rice that produces vitamin A called miracle rice or golden rice. A gene for beta carotene was introduced into this rice. This is yellow in color because of large amounts of beta carotene. Another example is flavor saver tomato. This tomato has a long shelf life because an enzyme called polygalacturonase is silenced in this tomato. The technique is called RNA interference. So this enzyme, which is involved in ripening, is not produced in this tomato. That is why it has a long shelf life. Another example is edible vaccines produced in banana. So in this banana, a gene for an antigen from a pathogen is introduced. And when this banana is consumed, it will produce antibodies against it in our body. Now let us look at some examples of genetically modified animals. The cow shown here is a genetically modified cow called Rosie. It produces the human protein called lactalbumin in its milk. Lactalbumin is a storage protein in humans. Another example is the sheep called Tracy. Tracy produces antitrypsin in its milk. Antitrypsin is used in the treatment of the lung disease emphysema. Another example of a genetically modified microorganism is genetically modified E. coli that produces the human protein insulin. It is called humulin. It was produced by the American company Eli Lilly and Company, USA. Another example of a genetically modified microorganism is Pseudomonas putida, which was genetically modified to degrade oil. It was created by an Indian scientist, Anand Chakrabarty. This bacterium was given the first biopatent. Now let us look at some examples which were created for merely academic interest. For example, a glowing cat, a glowing mouse, a glowing fish. All these animals have the protein called green fluorescence protein from jellyfish. We have even created a bioluminescent tobacco which has a gene from firefly. The gene is for the enzyme called luciferase. From these examples, it is evident that genetic engineering is very important. So I am repeating once more. Genetic engineering involves the isolation of a useful gene from one organism and insertion into another organism to produce a new combination of DNA called recombinant DNA. The first recombinant DNA was produced in 1972 by the scientist Paul Berg. He is called the father of genetic engineering. What he did was he inserted 3 genes of the bacteriophage called lambda phage into the DNA of the virus SV40. The first genetically modified organism was created by Stanley Cohen and Herbert Boyer in 1973. It was a genetically modified E. coli. What they did was, they introduced an antibiotic resistance gene, which is kanamycin resistance gene, from the bacterium Salmonella typhimurium into a plasmid. As you know, plasmid is an extra chromosomal circular DNA present in bacteria. This plasmid was then inserted into E. coli, in which the foreign gene was expressed. In other words, we can say this E. coli was genetically modified. It was able to grow on a medium containing the antibiotic kanamycin.
Hello, this is Dr. Jose Kudakasseril. I am going to talk to you today about the tools of genetic engineering.
We need two different tools in biotechnology or genetic engineering. One is DNA modifying enzymes. Three enzymes are required. One is restriction endonuclease that cuts DNA. Second is DNA ligase that joins DNA. Third is DNA polymerase that replicates DNA. Second tool is vectors. First, let us talk about restriction endonucleases. They are also called molecular scissors or chemical scalpels. They were discovered by Nathan Smith, who got Nobel Prize for this discovery. These enzymes cut DNA at specific locations. They are called recognition sequences. They are produced by bacteria to cut and inactivate the DNA of viruses. They do not cut bacterial DNA because the recognition sequence is methylated in bacteria. These recognition sequences are palindromes. A palindrome is a word or a sentence which can be read in both directions. Example, Malayalam, a man, a plan, a canal, Panama. The recognition sequence of EcoR1, which is an enzyme obtained from E. coli, is GAATTC. Now let us talk about the naming of the enzyme. For example, in EcoR1, E stands for Escherichia, which is the genus. CO stands for coli, which is a species. R stands for the strain, which is RY13. And 1 stands for the first identified restriction endonuclease in E. coli. These enzymes are of three types. Type 1, which cut DNA approximately 1000 nitrogen bases away from the recognition sequence. Type 2, which cut DNA at the recognition sequence. Because of their specificity, they are used in genetic engineering. EcoR1 is an example of a type 2 enzyme. Type 3, which cut DNA approximately 25 nitrogen bases away from the recognition sequence. Type 2 enzymes are again divided into two. Those that produce sticky ends, example EcoR1, and those that produce blunt ends, example SMA1. In the case of EcoR1, it cuts at the specific location shown by arrows, resulting in the production of sticky ends, also called cohesive ends or overlapping ends. In the case of SMA1, which is obtained from Serratia marcescens, it cuts the DNA right in the middle. So the ends of the DNA has blunt ends. The first restriction endonuclease to be discovered was HAND2 from Haemophilus influenzae. HAND2 always cuts DNA at a specific sequence of 6 base pairs. The second enzyme used in genetic engineering is DNA ligase. It joins pieces of DNA using energy from ATP. It was discovered by Esther Lederberg. It is present in all bacteria. The third enzyme is DNA polymerase that replicates DNA. It was discovered by Arthur Kornberg. Taq polymerase, which is a DNA polymerase obtained from bacterium Thermus aquaticus, is used in PCR or polymerase chain reaction, which I will talk to you in the next class. Thermus aquaticus is found in hot springs. Therefore, this enzyme can withstand high temperature. So, we have discussed three enzymes used in genetic engineering: restriction endonuclease that cuts DNA, DNA ligase that joins DNA, and DNA polymerase that replicates DNA.
Today, I am going to talk about one of the tools in genetic engineering, that is vectors. The reason for using vectors is that when you use vectors, 10% of the cells get transformed. If you don't use vectors, only 1% of the cells get transformed. Vectors are of two types. Number one, cloning vectors. Cloning vectors are used to make multiple copies of the gene. For example, in the second step of genetic engineering, which is called amplification of the gene, we are making multiple copies of the gene. Two commonly used vectors for that purpose are: one is PBR322, which was an artificial plasmid created by two scientists, Bolivar and Rodriguez, and PUC18, which was created at the University of California. Two vectors which were created for the Human Genome Project are bacterial artificial chromosome and yeast artificial chromosome. These are all cloning vectors. Cloning vectors are all artificial vectors. Number two, expression vectors. These are used to transfer genes into various organisms. In plants, the commonly used vector is TI plasmid or tumor-inducing plasmid of Agrobacterium tumefaciens. This bacterium infects only dicot plants and causes the disease crown gall tumor. Another example of an expression vector is retrovirus. Retroviruses are RNA-containing viruses. Example, murine leukemia virus is used to transfer genes into animals and humans. Genetic engineering in humans to cure genetic diseases is called gene therapy. Expression vectors are all natural vectors. Now let us look at the characteristics of a vector. Example, PBR322. As you can see in the picture, it has a region called ORI. It stands for origin of replication. That is where replication starts when this plasmid is put in E. coli. Or in other words, we can say that is a place where DNA polymerase of E. coli binds to make multiple copies of the plasmid. Second one, selectable markers. These genes are actually used to identify cells or E. coli cells which contain the foreign gene. For example, in PBR322, there are two marker genes which are antibiotic resistance genes. One is ampicillin resistance gene and the other one is tetracycline resistance gene. Ampicillin resistance gene produces an enzyme that breaks down the antibiotic ampicillin. Similarly, tetracycline resistance gene produces an enzyme that breaks down the antibiotic tetracycline. So these are required to select E. coli cells which contain the foreign gene, or in other words, we can say transformant bacteria. Near to the ORI region, you can see a region called ROPE. The full form of ROPE is repressor of primer. It produces proteins involved in the replication of the plasmid. In other words, the number of copies of the plasmid in E. coli is determined by the region called ROPE. If you modify ROPE, you can increase the number of copies. Normally, if you talk of selectable markers, genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, kanamycin, etc. are used as selectable markers in E. coli. The normal E. coli cells do not carry resistance to any of these antibiotics. Now, the third character of a vector is what are called cloning sites or restriction sites. These are places where different restriction endonucleases cut the DNA. For example, you can see a lot of places on PBR322 where different enzymes cut the DNA. They are called cloning sites or restriction sites. But one thing to remember is that one enzyme should have only one site, because If an enzyme has more than one site, the plasmid will be cut into several pieces. The plasmid has to maintain the circular shape. Therefore, there should be only one cloning site for one restriction endonuclease. I'll repeat once more. A vector should have an origin of replication or ORI, selectable markers for transformation, and several cloning sites or restriction sites for several restriction endonucleases, but one enzyme should have only one site.
Today I am going to talk about the steps in genetic engineering.
The first step in genetic engineering is isolation of the gene. Usually, we isolate genes from a cell culture. For example, if you are going to isolate a human insulin gene, you have to first make a human cell culture. Second step is called amplification of the gene. There, we are making multiple copies of the gene. The old technique is called gene cloning in bacteria, where we use cloning vectors such as PBR322 and PUC18, details of which I am going to talk in the next video. Second technique is called polymerase chain reaction or PCR. There we use a machine called the thermal cycler. It is a very fast technique in which within 90 minutes we can make 1 billion copies of the gene. So it is a much better technique compared to gene cloning in bacteria. Third step is insertion of the gene into an organism. There are two methods. One is direct gene transfer. That does not use a vector. For example, gene gun method does not use a vector. You have to remember one thing. Direct gene transfer is not preferred because it results in only 1% transformation. The second one is indirect gene transfer using a vector. For example, in plants, the vector used to transfer genes is the Ti plasmid of Agrobacterium tumefaciens. Fourth step in genetic engineering is screening of transformed cells. There, what we are doing is using a marker gene, we identify the cells which contain the foreign gene. We can either use antibiotic resistance genes as marker genes, or you can use green fluorescence protein gene, which is obtained from jellyfish, as the marker gene, and luciferase gene from firefly as the marker gene. So these are the four steps in genetic engineering. Now let us look at the first step in genetic engineering, isolation of the gene. In this step, what we are doing first is we are isolating DNA from a cell. If you are isolating DNA from a plant cell, first you have to remove the cell wall using enzymes such as cellulase, hemicellulase, and pectinase. If you are isolating DNA from a fungal cell, you use chitinase. If it is a bacterial cell, we use lysozyme. Then using protease, we remove proteins in the cell. In the next step, using ribonuclease, we remove RNA. Then we add chilled ethanol or isopropanol when DNA precipitates. This precipitated DNA comes to the top. It is pooled on a glass rod. This is in the picture you can see the isolated DNA. The isolated DNA is then cut with a restriction endonuclease when hundreds of DNA fragments are obtained. Then we separate the DNA fragments based on their size and charge differences using a technique called gel electrophoresis.
Today I am going to talk about one of the important techniques in biotechnology, namely gel electrophoresis.
Gel electrophoresis is a technique by which DNA, RNA, or proteins are separated in a gel on the basis of size and charge differences. It was invented by the scientist Teselius. If you are separating nucleic acids such as DNA and RNA, an agarose gel is used. But if you are separating proteins, a polyacrylamide gel is used. Agarose is a carbohydrate polymer obtained from red algae such as Gelidium and Gracilaria. When agarose is heated with water and cooled, it becomes a gel. The gel has a mesh-like appearance with holes in it. When DNA fragments go through the holes, the small fragments can easily go through the holes, whereas the large ones cannot. That is how DNA gets separated in the gel. The concentration of agarose used depends on the size of the DNA fragments to be separated. For example, we usually use concentration between 0.5 and 1%. The gel electrophoresis setup has the following parts. It has a gel tank with two chambers in it, in which we take buffer, a casting tray in which we make the gel combs, which are plastic devices by which you can make holes in the gel called wells, the electric wires, and the power supply. To make the gel, agarose is mixed with water and heated till it melts. It is then poured into the gel tray. When it cools, it becomes a gel. A comb is put in the gel vertically to create the wells. The wells look like this. We apply the DNA samples inside the wells. DNA is mixed with a tracking dye called bromophenol blue and glycerol before applying to the wells. This dye is called a tracking dye because it tracks the movement of the DNA fragments through the gel. The dye moves in front of the smallest DNA fragment. In the first well, we add a DNA sample containing DNA fragments of known molecular weight to create a DNA ladder. Other DNA samples are applied in the other wells. A DNA ladder is used to determine the size of an unknown sample. It contains regularly spaced DNA fragments of known sizes. When run on an agarose gel, it looks like a ladder. Buffer is added to the two chambers. A buffer is added because it provides ions that carry a current through the gel. It also maintains the pH inside the chamber. The apparatus is then connected to a power supply and current is passed through the gel. DNA is negatively charged because of phosphate in the DNA, so the DNA fragments will move towards the positive electrode or anode. The smaller the DNA fragment, the further it moves. The dibromophenol blue moves in front of the smallest DNA fragment. So the largest fragment will be near the wells and the smallest fragment will be near the anode. After electrophoresis, the DNA fragments are visualized under UV light by staining with ethidium bromide. Ethidium bromide is a fluorochrome. A fluorochrome is a chemical which absorbs UV light and emits visible light. This dye gets into the grooves of DNA. DNA appears as orange bands when seen under UV light. A band in the gel consists of many DNA fragments of the same length. As you can see in this picture, the bands near the well are thicker than the other ones. This is because the DNA fragments there are of the largest size. The one at the bottom is the smallest size. In other words, one band contains many DNA fragments of the same length. So all the bands will be of the same width. Gel electrophoresis is used not only in genetic engineering but also in DNA fingerprinting. In DNA fingerprinting, it can be used to identify criminals. For example, in the picture here, the crime scene DNA matches with the suspect 2 DNA. So suspect 2 is the criminal. Gel electrophoresis in DNA fingerprinting can be used to solve paternity disputes. In the picture here, the DNA of male 1 matches with the DNA of the child. So he is the real father of the child. In this video, I am going to talk about the second step in genetic engineering, namely amplification of the gene. Two techniques are used. One is gene cloning in bacteria, where we use cloning vectors And the second technique is polymerase chain reaction. Today, I am going to talk about how we clone genes using the cloning vector PBR322. PBR322 is an artificial vector which is used for cloning of the foreign gene in E. coli. It was created by two Mexican scientists, Bolivar and Rodriguez. PBR322 has the following features. It has an origin of replication called ORI, that is where replication starts to make multiple copies of the plasmid in E. coli. The next character is the ampicillin resistance gene or AMPAR, which codes for an enzyme that breaks down the antibiotic ampicillin. The next character is the tetracycline resistance gene, which codes for an enzyme that breaks down the antibiotic tetracycline. Both of these are called marker genes because they are used to identify E. coli cells which contain the foreign gene. PBR322 has 40 cloning sites or restriction sites. These are places where different restriction endonucleases cut the plasmid and we can insert the foreign gene in one of those sites. The other character is the rope, which is the full form is repressor of primer. That rope determines how many copies of PBR322 are produced in E. coli. The usual number is 20 copies. If you modify rope, you can increase the number of copies. The actual picture of PBR322 shows all the 40 different cloning sites. These sites are also called restriction sites. Now, in PBR322, we usually insert the foreign gene inside the tetracycline resistance gene. BAM-H1 is a restriction endonuclease that cuts inside this gene. BAM-H1 is obtained from the bacterium Bacillus amyloliquefaciens. When the gene is inserted inside the tetracycline resistance gene, the tetracycline resistance gene becomes inactive. It does not produce the enzyme anymore that breaks down tetracycline. This is called insertional inactivation. In the procedure, we take along with the buffer PBR322 restriction endonuclease that cuts inside the tetracycline resistance gene, the foreign gene and DNA ligase. Here, what happens is that some of the PBR322 take up the foreign gene, others will not. So we get two types of PBR322, those with the foreign gene and those without the gene. Then these PBR322 are incubated with E. coli. We give E. coli a heat shock treatment in which we heat E. coli to 42 degrees centigrade and suddenly cool it to 0 degrees centigrade. We also give a calcium chloride treatment. These treatments increase the permeability of the E. coli membrane so that PBR322 can go inside E. coli. Some of the E. coli will take up PBR322, others will not. So we get three types of E. coli. First one is called the non-transformant, which has not taken up the PBR322. The other two are transformants. But the first transformant is called the non-recombinant because it has only PBR322 in it. It does not have the foreign gene. The third one is the recombinant bacteria, which contain PBR322 and a foreign gene. We are interested only in the recombinant bacteria. So let us see how we select out the transformant, or we can say recombinants, from the rest of the bacteria. E. coli cells are cultured on potato dextrose agar medium, which is the usual medium used to culture bacteria, containing the antibiotic ampicillin. The E. coli cells divide and form colonies. Each colony will have thousands of cells from in it, all derived from one E. coli. The colonies are white in color and they project from the medium like this. Just remember, each colony will have only one type of E. coli in it. Because of the presence of ampicillin, non-transformant cells will die on that medium because it does not have PBR322 in it. PBR322 has the ampicillin resistance gene. Because of the absence of that gene, the non-transformants will die on that medium. This petri dish is called the master plate. Now, the transformants which are growing on the medium are transferred to another medium containing tetracycline. The technique is called replica plating. In replica plating, we take a wooden disc, attach it to a handle, cover the wooden disc with velvet cloth, and it is sterilized in an autoclave to kill all the bacteria on the velvet cloth. It is then put on the top of the petri dish so that the velvet cloth touches all the colonies. Remember, all the colonies are projecting upwards. So few cells from each colony will attach to the velvet cloth. This device is then put on another medium containing the antibiotic tetracycline. On the tetracycline containing medium, recombinants will die because when the foreign gene is inserted inside the tetracycline resistance gene, that gene becomes inactive and it does not produce the enzyme that can break down tetracycline. So they will die on that medium. Those which survive on that medium are the non-recombinants. This petri dish is called the replica plate. Now we compare the master plate and the replica plate. As shown in the diagram, two colonies shown in yellow color found in the master plate are missing in the replica plate. Those are the colonies we are interested in. They are selected from the master plate. Then with a spatula, we take out the colonies and culture them so that when E. coli multiplies, PBR322 multiplies and the foreign gene multiplies. So we get large quantities of the foreign gene. This technique is called gene cloning in bacteria. Today I am going to talk about how we can clone genes using the vector PUC18. In the last class, I told you about how we can clone genes using PBR322. Here I am going to talk about another cloning vector called PUC18, which stands for Plasmid University of California 18. It is one of the most widely used cloning and expression vectors. It was produced by a modification of PBR322. PUC8 has a gene for antibiotic resistance, AMPR. It has a gene called the lac acid gene and its promoter, which produces the enzyme beta-galactosidase. Lac acid gene is one of the genes in the lac operon. This gene contains a polylinker region or a region where several restriction endonucleases can cut the DNA. And the rope region here is absent because we are trying to increase the number of copies of PUC18. Remember I told you ropes determines the number of copies of the plasmid produced in E. coli. In the case of PBR322, it was 20 copies. Here it is 500 copies. The foreign gene is inserted in the lac acid gene, leading to its inactivation, and when it is inserted, the enzyme beta-galactosidase is not produced. This is called insertional inactivation. In the procedure, we incubate PUC18 along with the restriction endonuclease that cuts in the polylinker region, the foreign gene, and the enzyme DNA ligase. Here what happens is that some PUC18 take up the foreign gene, in others it will not. So we get two types of PUC18, those with the foreign gene and those without the gene. Those two types of PUC8 are incubated with E. coli. In order to make the membrane of E. coli permeable to PUC18, we give E. coli a heat shock treatment in which we heat E. coli to 42 degrees centigrade and suddenly cool it to 0 degrees centigrade. We also give a calcium chloride treatment. Calcium ions increase the permeability of the membrane. So what happens here is that E. coli cells take up PUC18, but only some cells will take up PUC18, others will not. So we get three types of E. coli. One in which There is no puck18, which is called the non-transformant. The other two are transformants. Of these, one has puck18, but it does not have the foreign gene, which is called the non-recombinant. And in the last one, which is the recombinant bacteria, it has the puck18 and the foreign gene. Remember, we are interested only in the recombinant bacteria. In order to select this bacteria, they are grown on a medium which is called the potato dextrose agar medium, containing ampicillin, which is an antibiotic, and a chromogenic substrate called X-gal. The full form of X-gal is 5-bromo, 4-chloro, 3-indole beta-D-galactose. The enzyme beta-galactosidase converts X-gal into a blue-colored compound. Now, what happens here is that the presence of the foreign gene inactivates the lactic acid gene in the recombinant bacteria. So, the conversion of X-gal into the blue-colored compound cannot occur, and these colonies will be white. Whereas, in the non-recombinant colonies, lactic acid gene is active, beta-galactosidase is produced, and the conversion to the blue-colored compound occurs, and they are blue in color. So, we select out the white colonies. This is how we use puck18 to clone the foreign gene. Today I am going to talk about one of the techniques used in amplification of the gene, namely polymerase chain reaction. This is one of the very important techniques used in biotechnology. Polymerase chain reaction, or PCR, was invented by Karamolis. He got Nobel Prize for this invention. PCR is commonly used in laboratories to make millions or billions of copies of a particular region of DNA. So here we are making millions of copies of the foreign gene, which is called gene cloning. Let us look at the requirements of PCR. We need a thermal cycler, which is a machine used for PCR, DNA to be amplified. In our case, it is the foreign gene. Then we need Taq polymerase. Taq polymerase is a DNA polymerase which is obtained from the bacterium Thermus aquaticus. This bacterium is found in hot springs. Therefore, the DNA polymerase of this bacterium can function at high temperature. We need 4 types of deoxyribonucleotides, or the nucleotides found in DNA. We need DNA primers. These primers are single-stranded DNA, around 20 nitrogen bases long, which are complementary to the 3' ends of the DNA. We use primers because Taq polymerase cannot initiate replication from the ends of the DNA. It can only extend a primer. We need 2 primers because the 2 strands of DNA go in opposite directions. One is 3' to 5', the other is 5' to 3'. These ingredients are assembled in a tube and put through repeated cycles of heating and cooling that allow the DNA to be amplified. Now let us look at the steps of PCR. PCR has 3 steps. First step is called denaturation, in which DNA is heated to 95 degrees centigrade to separate the 2 strands of DNA, or denature the DNA. In the second step, DNA is cooled to 55 degrees centigrade. That is when the 2 DNA primers that we have synthesized bind to the 3' ends of the DNA. This is called annealing. In the third step, which is called primer extension or chain elongation, we heat the DNA to 72 degrees centigrade, which is the optimum temperature of Taq polymerase. Taq polymerase binds to the 2 primers and extends the DNA in 2 directions, or in other words, replication occurs. So after replication, we get 2 copies of the original DNA. This is called one cycle of replication. This is repeated again and again till millions of copies of DNA are obtained. After 30 cycles of replication, which takes 90 minutes, we get 1 billion copies of DNA. PCR has many research and practical applications. It is routinely used in DNA cloning. That is, in genetic engineering, we clone the foreign gene before insertion into an organism, which is also called amplification of the gene. In the second application, we use PCR to find out the presence of a pathogen in the host. For example, in COVID-19 testing, we use PCR to find out the presence of the coronavirus in the host. Third, forensic analysis of DNA, or in other words, in DNA fingerprinting, the DNA present at the crime scene is so small in quantity, we have to amplify that DNA using PCR. Now let us look at RT-PCR, or reverse transcriptase PCR, which is the technique used in COVID-19 testing. Coronavirus, which causes COVID-19, is a retrovirus or an RNA-containing virus. From the RNA of this virus, a complementary DNA is produced using reverse transcriptase. This DNA is then amplified using PCR. So PCR is a very important technique in biotechnology with a lot of applications. In direct gene transfer, since we are not using vectors, only 1% of the cells get transformed. Before we do direct gene transfer, the foreign gene is linked to a marker gene. The marker gene identifies the cells which contain the foreign gene. The marker gene used in plants is either kanamycin resistance gene or Gus gene. In animals, neomycin resistance gene can be used. Gus gene produces an enzyme beta-glucuronidase that converts X-gluc into a blue-colored compound. X-gluc is similar to X-gal. The full form of X-gluc is 5-bromo-4-chloro-3-indolyl beta-D-glucuronic acid. When Gus gene is used as the marker gene, all the transformed cells will be blue in color. Now remember, Gus gene is also an example of a reporter gene. A reporter gene produces an enzyme that catalyzes a color reaction by which transformed cells can be identified. In the case of plants, we use protoplasts for direct gene transfer. Protoplasts are plant cells without a cell wall. The cell wall is removed using enzymes such as cellulase, hemicellulase, and pectinase. There are 5 methods of direct gene transfer. Number 1, microinjection. Number 2, chemically mediated uptake using polyethylene glycol. Number 3, electroporation. Number 4, bioelistics or gene gun method. Number 5, liposome mediated transfer. First, let us look at microinjection. This method is mostly used in animal cells. Using a micro syringe, the DNA solution is directly injected into the nucleus of the cell. Once it is inside the nucleus, the foreign gene and the marker gene insert into the DNA of that organism. The second method is called electroporation. The material to be transformed is incubated in a buffer solution containing the foreign gene and the marker gene. It is subjected to high voltage electric pulses. This induces holes in the plasma membrane through which the foreign gene enters the cell and integrates into the genome. Third method uses polyethylene glycol. Using polyethylene glycol and divalent cations such as calcium ions, we can introduce the foreign gene into the plant cells. These chemicals destabilize the plasma membrane and produces holes in it through which the foreign gene can enter the cell. Once it has entered, the DNA integrates into the genome. Fourth method is called gene gun method or bioelistics. Using a gene gun, Microscopic bullets, which are either made of tungsten or gold, which are coated with the foreign gene, are fired into the cells. This technique is called biolistics, which is used only in plants. The fifth method is liposome-mediated transfer. Liposomes are circular lipid molecules with an aqueous interior that can carry nucleic acids. It encapsulates the DNA fragments. It adheres to the cell membranes, fuses with the membranes, and transfers the foreign gene into the host DNA. So these are the 5 methods of direct gene transfer. Remember, direct gene transfer is not preferred because it results in 1% transformation, whereas if you use vectors, it results in 10% transformation. The vector which is used to transfer genes into plants is the TI plasmid or tumor-inducing plasmid of Agrobacterium tumefaciens. In animals, we can use DNA of retrovirus, example murine leukemia virus. Retrovirus contains RNA as a genetic material. From the RNA of this virus, a complementary DNA is produced using reverse transcriptase. This DNA is used as a vector. Now let us look at the TI plasmid. Agrobacterium tumefaciens is a gram-negative bacterium that lives in the soil. It causes a disease in dicot plants called crown gall tumor. The bacterium is attracted to wound tissue of dicot plants since it produces a phenolic compound called acetosyringone. This compound is absent in monocots. That is why Agrobacterium does not infect monocots. The bacterium attaches to a plant cell and transfers a small portion of the TI plasmid called tDNA, the full form is transferred DNA, into the plant cells. This tDNA is incorporated into the plant DNA, or we can say the plant cell is transformed. The tDNA contains 3 genes. One is for the synthesis of the plant hormone auxin and cytokinin, and for compounds called opines. The transformed plant cell produces large amounts of auxin and cytokinin, which are cell division inducing hormones. So this plant cell divides repeatedly to form a tumor. The opines are actually amino acid analogs used as food by the agrobacterium. Since the formation of the crown gall tumor is an example of genetic engineering occurring in nature, Agrobacterium is called the natural plant genetic engineer. To use TI plasmid as a vector, it is disarmed by removing the genes from the tDNA. In that location, the foreign gene and the marker gene, we can use kanamycin resistance gene as the marker gene, or we can use Gus gene as the marker gene. This modified TI plasmid is then inserted into Agrobacterium, which is used to infect plant cells. The method used to transfer genes into plants is called the leaf disc transformation method. This method involves plant tissue culture. So all the techniques have to be done under sterile conditions. First, we take leaves of dicot plant to be genetically modified. We surface sterilize the leaf we can use chemicals such as 70% ethanol, 10% calcium hypochlorite, or 0.1% mercury chloride. Small sections called leaf discs are cut from the leaf. These leaf discs are put in a suspension of Agrobacterium. Since the wounded tissue of the leaf disc produces acetosyringone, Agrobacterium is attracted to the leaf disc and they transfer the tDNA into the plant cells. Now remember, the tDNA contains the foreign gene and the marker gene. So in order to select the cells which contain the foreign gene and the marker gene, the cells are cultured in a tissue culture medium containing kanamycin and an antibiotic called carbapenicillin. The purpose of using carbapenicillin is to kill Agrobacterium, which may be sticking to the leaf discs. Kanamycin is added to the medium because our marker gene is kanamycin resistance gene. So here what happens is that all the cells which are transformed, which contains the kanamycin resistance gene, will survive on this medium. all the other cells will die on the medium. In order to make plants from the surviving cells, this medium contains high concentration of cytokinin and low concentration of auxin. That ratio induces shoot formation. To make roots from the shoots, they are transferred to a medium containing auxin. So this results in root formation, and these plants are then taken out, they are hardened and then transferred to polythene bags or pots. The plant that we have created can be called a transgenic plant or a genetically modified plant. To summarize all the steps in genetic engineering, first step is isolation of the gene. Second step is amplification of the gene. We discussed two methods. One is gene cloning in bacteria using the cloning vectors PBR322 and PUC8. A better technique is polymerase chain reaction or PCR. In the third step, we discussed insertion of the gene into an organism. We discussed direct gene transfer and indirect gene transfer. In the fourth step, we selected out the transformed cells using the marker gene, that is kanamycin resistance gene. So these are the 4 steps of genetic engineering. Thank you for listening to my video.
I have published some books for students appearing for the NEET exam. One is Handbook of Biology, Volume 1, which describes all the chapters in class 11, and Volume 2, which describes all the chapters in class 12. And also I have published a book, Biology in a Nutshell, which includes all the chapters. These books are available on Amazon. I am giving the link below.